Hi, good afternoon to everyone. I'm Ivy Rom from Divisions of Program Promotion and also moderator for today's webinars. Welcome to joining us this afternoon. For your information, today's webinar will be divided into topic sharing about 45 minutes and Q&A sessions about 15 minutes. If you have any questions, please paste at the chat box. We may not answer all the questions if there is time constraint. Today's our topic is vaccines and immunization, battles against infectious disease. Before we start, let me introduce our speaker, Dr. Tan Kim Chong. Dr. Tan is Utah Assistant Professor from Faculty of Science, Head of Department from Departments of Allied Health Sciences. Dr. Tan obtained his Bachelor's of Health Science, Honorable Major Biomedical, and further study his doctorate program, Doctor of Philosophy, major in vaccinology in University Science Malaysia. His areas of expertise is medical microbiology, molecular biology, and vaccinology. Without any further delay, let's welcome our speaker, Dr. Tan.
Okay, thank you for kind introduction from our colleague Ivy there. Okay, just some additional to what she have mentioned just now. Okay, I graduated from USM and I work on vaccine. And you might want to ask some of the point that she mentioned just now. I also work on molecular biology and also some other fields, like for example, molecular pathogenesis. Okay, all of this actually is part of the journey of studying vaccine. Okay, so my journey of studying vaccine starts from molecular biology, where we study on the genome. Then we did some modification to the genome, and we create a live attenuated vaccines. Then we do some animal study, and we study on the immune response towards the vaccine candidate that we have created. So in later on the presentation that I share with you all, I will actually bring you all through some of the explanation on some different types of vaccines that are currently available in the market, and what are the differences between those vaccines and how these vaccines help in preventing infectious disease. Especially now, we are actually under the threat of this COVID infection, and with the current COVID vaccine that are available, so a lot of individuals, they actually worry about getting vaccinated, and also they are worried about the side effect of vaccination. So throughout the presentation, if you have any question, you can actually post it, and I'll try my best to answer you all. So Ivy, can I share slides? Yeah.
Can
Okay. So yeah. So this is my presentation. So basically, I will be focusing on some of the basic information, because it is very important, I think, for the public to understand the basic things about immunization and vaccination. Because in the Facebook, in the media, in the social media, there's a lot of information. Partly, mainly, they are all unverified, not verified information. So it is very important to provide you all with accurate and important information, so that you will to clear your doubt and concern about vaccination. So what you can see here is uh, vaccine is very important for the battle against infectious disease. Without vaccine, a lot of disease, a lot of infectious disease could actually uh, take away life. Okay, take away life when an individuals get infected. So with the presence of vaccination, we can actually prevent the occurrence of a lot of disease. So in this topic that we will look into today, we actually will cover the topic on infectious disease. What are the different infectious diseases that are currently threatening the health of human being? And I also introduced to you all some of the emergence or emerging diseases, okay, in the past and in presence, okay, vaccination and immunity, the importance of it and how it developed. And I think a lot of you are interested in types of vaccine. Okay, so even for the COVID, you will notice that there are a lot of different types of vaccine available for COVID. Okay, the same goes with other types of pathogens. Okay, other bacteria or viruses that causes diseases. So I will also give you all some brief introduction on vaccination schedule. I think a lot of parents, if parents are some of the audience here, you know that once a baby is born, they will go through several schedule of vaccination. Okay, this is actually to protect the baby. Okay, then I will briefly bring you all through some of the COVID vaccine, okay, that are available. Then I'll try to answer some of your question. Okay, so to introduce your COVID vaccine, no, the infectious disease actually can be caused by many uh, many pathogens. So such as bacteria, virus, or parasite and fungus. So these are a few important organisms that could lead to disease state in an individuals. Whether you can get it directly or it has to you can get it indirectly. Okay, so those indirect approach normally it is spread by vectors. For example, the dengue virus, the malaria virus, okay, those malaria parasites, okay, those are spread by mosquito. Okay, so for bacteria, it can actually spread through consumption of food or drinking of water contaminated by bacteria, or it could also enters into your body and cause disease through a simple needle prick. Okay, those can be caused by bacteria that reside at the uh, needles that enters into your bloodstream and leads to a disease state. So there are many ways infectious pathogens can enter into our body and lead to a disease state. Okay, some is even caused by this human body fluid contact, for example, the HIV virus. So all of these uh, they have different means of spreading, and by understanding the pathogens, by understanding the organisms, and by understanding the way it spread, it helps us to control the spread of the disease. Just like what we have here now, what we understand about COVID, we know that it is spread through airborne, and it can be prevented by wearing masks, and we can also reduce the risk of spreading by constantly washing our hands with soap and sanitize it with alcohol-based disinfectant. So these are ways that we understand the disease, understand the spread of disease, and how we can control it. Okay, so in the event of cases where uh, if you don't understand the disease, the spread of the disease could, okay, the spread of the infectious disease could actually go beyond our control. Okay, so another thing that I would like to highlight here is that a lot of time the infectious disease, okay, infectious pathogens, they might not lead to a disease state. However, in individuals that are immune compromised, okay, so for example, the AIDS patients, their immune system are compromised, the baby, their immune system are not well developed, the elderly, their immune systems are weak. So those individuals actually they can get infected easily. Okay, they can get infected easily. That's why later on we will actually cover another topic briefly also on herd immunity. And I think all of us are talking about that nowadays. Even the government every day in the news they are actually talking about herd immunity. So we also will look into that on how these weakened individuals, okay, immune weakened individuals can be protected through herd immunity later. Okay, so under normal condition, our immune system will work perfectly well and they will be able to uh, counter the attack by the pathogen. However, if the pathogens produce toxins and have some pathogenic component excreted by the pathogens, we might not able to survive. Okay, so that's why vaccine is actually very important in this way, in this counter or battling against infectious disease. Okay, in some condition, for example, without the BCG, actually the tuberculosis progress and infect many of the population and causes a lot of death. Okay, this is how vaccine improve the uh, well-being of individuals, human being in the world. Okay, So another important thing is that vaccine helps to prevent the disease. Okay, vaccine help to prevent the disease. Okay, a lot of bacteria, especially bacterial pathogens, when you get infected, okay, nowadays we treat that infection with antibiotics, and we also know that a lot of bacterial pathogens, they are drug resistant. So if that is the case, most of the time the individuals that are infected by drug resistance pathogen, they will not survive. Okay, that's why it is very important if we can vaccinate the individuals, and the individuals has the immune response against the pathogen, and the disease will not deteriorate and will not get worsened, and the individuals will recover speedily. Okay, so that is about infectious disease. Okay, okay. So before we go further, I just want to give you some brief introduction on infectious disease. I use the example of cholera pandemic. Okay, just for information, actually I work on a live attenuated vaccine for cholera. So I use this example for you to understand better how pandemic happen. Okay, pandemic, normally we talk about pandemic is when a disease start at the local area and is spread to the surrounding and worldwide. So this will lead to a pandemic, occurrence of pandemic. And most of the time when pandemic happens, it kills a lot of life. So for example, here we can see cholera. So there are total of 7 pandemic that we can see here, and 6 of it is actually caused by Vibrio cholera 01 sero group. Okay, and the 7 is Altor classical, 01 Altor class, 01 Altor, and the first 6 is 01 classical. So and after that, we also found that there are emergence of Vibrio cholera 0139 Bengal strain in 1992, also kills a lot of individuals in Indonesia and Southeast Asia. So this is the spreading of a disease without the control of the human movement. Again, nowadays we have a lot of flight shipment. So the control of human mobility is very important in curbing the spreading of the disease. That's why when we bring back the example of COVID, we can see that with the advancement of this traveling mobility in our aviation system, that we can just fly here and there within few hours time, we can be in another city or country. So the control of the spread of the disease will be very limited. So if we don't control human mobility, the spread of the disease will also be very rapid. Okay, so since I used the example of cholera, okay, now we look into the genetic of it. Okay, nowadays when we talk about infectious disease, the spread of diseases, the pandemic, we will look into a very fine detail at the molecular level, which is the gene level. Okay, the gene actually can tell us a lot of things. The gene can tell us whether the disease, okay, the origin of the disease, where the strain come from. Okay, by looking at the genetic, we can check the ancestry of the strain of the virus or bacteria. So what we can see here, the genetic of this cholera, how this cholera become pathogenic, become infectious, become deadly when it infect human. Okay, so originally it is not deadly, but however, with the acquiring of external gene, a toxin gene from a CTX phage, a virus that infect the bacteria, they acquire this toxin gene, and this toxin gene is the one that increases the pathogenicity, increases the virulence, increases the danger of this Vibrio cholera bacteria. Okay, so without this CTX phage, without this cholera toxin, basically this Vibrio cholera will not lead to diarrheal state. Okay, so I want you all to actually remember this, because later on we will also I will also bring to you some of the evolution part, okay, evolution part of a pathogen. Okay, So originally a pathogen that might not be dangerous to human, eventually it will develop into a dangerous form of virus. Okay, there are also hypotheses stating that the COVID, okay, the COVID strain of virus originally it doesn't kill. Okay, it is not as deadly as it is now. Okay, however, through evolution, through mutations, it acquires some ability to kill human. Okay, ability to cause more uh, detrimental effect when it infect human. Okay, so now we go into the history of vaccine development. So vaccine development starts actually very simple. Okay, with this discovery of this uh, infection of uh, non-related, okay, non-related pathogen that brings some protective immune response, okay, protective immune response towards deadly pathogens. Okay, so Edward Jenner found that this cowpox doesn't cause any deadly condition in human. It just cause some fever and some mild complication. Okay, it is not deadly. And those individuals, okay, immunized or infected by cowpox, they become immune towards smallpox. Okay, so because the sharing of antigens, sharing of component in cowpox, okay, that immune response that develop against cowpox are able to protect against smallpox. This is how the vaccine get developed, and they start to study more and more on the vaccine. Okay, then we can see the BCG. Actually, BCG strain is not a TB strain. Okay, it is not a tuberculosis strain, but it is a closely related strain to tuberculosis. So when you get BCG vaccination, okay, the TB, you will be protected against the TB also. So here what we are looking at live attenuated vaccine here. Like for example, BCG is actually a live vaccine. So if you can recall, you can touch your arm there. If you can feel the bulging of this BCG vaccination still available, meaning that your BCG strain, the bacteria still present and still actively inducing your immune response. Okay, this is about BCG. And we have the development of vaccine as it progress, then starting from the live vaccines, they go into epitope vaccines, they go into dead vaccines, and many more. Okay, so advancement in the application of molecular genetic, the knowing or understanding of this gene component that presents. Okay, previously in the past, they do not know so much about genes. Now they have more information with the advancement of sequencing. They know about genes. They know about the amino acid sequence. Okay, they also know that how these proteins work in immune response. So they develop and they study with vast information, vast knowledge generated. They apply it in vaccine development. Okay, so for example, you have this recombinant hepatitis B vaccines. Okay, you have this DNA vaccines developed. Okay, viral vectors vaccine. Okay, uh, DNA vaccine, I think you're more interested because it is very related to the current application of COVID vaccine. Okay, and also viral vector vaccine. It is actually very related to what we are concerned now about the COVID vaccine. So we will look into that. And one very important thing is that the safety and efficacy of the vaccine also is of our concern. Okay, the safety and efficacy of the vaccine is very important because we don't want to create a vaccine that actually cause damages, okay, cause death. Okay, we want to create a vaccine that is effective and it is protective against individuals. Okay, so talking about vaccination, it is actually very simple, safe, and effective ways to protect individuals from harmful disease. Okay, imagine that I just give you a very simple example. Okay, uh, now we have this chickenpox vaccine. Okay, now we have chickenpox vaccine, and if you get vaccinated, okay, doesn't mean that you won't get chickenpox. You might still get a chickenpox depending on the immune response that it is induced. But it will actually reduce the severity of chickenpox. Don't think that chickenpox will not lead to death. Chickenpox might have a chance of leading to death if the individuals are immune compromised or immune systems are very weak. Okay, so with vaccination, you can actually reduce the effect, okay, the impact or the clinical symptoms of chickenpox. You can actually increase the rate of recovery in individuals. So this is very effective ways of controlling the disease. Okay, for example, BCG. Once you get BCG, you get sort of protected against the Mycobacterium tuberculosis. You know, when an individual get a TB infections, they have to go through a series of treatment, 6 to 12 months of antibiotic therapy. Okay, so this, if the strains that they get infected is a very virulent one, it might lead to death. So vaccination actually prevent the disease, harmful disease, from attacking those immunized individuals. They also reduce the symptom, okay, because most of the time the symptoms is the factor that leads to death of the infected individuals. Okay, so vaccination uses your own immune system to create antibodies that helps to protect you from the subsequent infection. Okay, uh, doesn't mean that, okay, vaccination doesn't mean that you, after you vaccinate, you won't get infected. Some of the vaccination is just actually enhance your immune response to battle against the pathogen attack. Okay, you might develop some of the sign and symptom, but you will not go to the extent of death. Okay, so vaccine train your immune system to create antibodies. Okay, so vaccine can contain killed or weakened form of germs like virus and bacteria or antigens. Okay, so we will go into this point later when we go into types of vaccine. Okay, vaccine do not cause the disease or put you at risk of its complication. Okay, vaccines save lives of up to 3 million people every year. This is the statistic given by WHO. So without vaccine, we can see up to 3 millions of individuals, human life, die of infection. Okay, so now we come into this herd immunity. Okay, if you remember just now, I did mention about the protection, okay, for those that are weak. Okay, if you refer to the example of COVID, you know that COVID is not given to babies, infants, or even adolescents. The young the young kids, they are not getting it. The old and diseased individuals, okay, also not getting the COVID vaccine. So how to protect these individuals? So we are actually immunizing the healthy one. Okay, make sure that the virus doesn't spread in the community. So if you are immunized, means the virus cannot infect you. So when you go back to your parents, okay, you are not bringing the virus to your parents, the elderly, or you're not bringing back the virus to your kids, which are not immunized. This is how herd immunity protect. Okay, herd immunity minimum, you need to have roughly around 70 to 80% of the population get immunized. Okay, so it reduces the risk of spreading in herd immunity. Okay, so in vaccination, the principle is actually very simple. So you get injection, it can be oral, okay, you can take it orally, okay, it can be mucosal, it can be even eye drops, nose drop, okay. Most of the vaccination that we see nowadays is through injection, okay, it's through injection. Okay, so injection, it will inject the pathogens, the antigens, or components of the virus or bacteria into your cells. Because all of these components that they inject into your body will treat them as foreign, okay, as something that is not from the body. Okay, so whatever that is not from the body, whatever that is foreign to your body, the immune system will treat them as enemy. Okay, so you will have these dendritic cells here, you have this macrophage here, will go and attack it and present those antigen, okay, those viruses to your immune system, and your immune system will create antibodies to tackle it. So the first round, when you get injected, your immune system will produce antibody. When second time, okay, most of the time, the second time will be the booster. You get injected again, it boosts the immune response by producing more of the antibodies. Okay, so when you really get infected by the pathogen, when the pathogen enters into your body already have abundance of antibodies, and it will actually inactivate and kill off the bacteria or virus. So this is how your immune system responds towards the infectious disease. So when your body is already prepared with antibodies that can kill the virus or bacteria, so when they attack your body will be safe and you will not get the disease. Okay, this graph itself is actually a typical explanation of why most of the vaccine you get booster. Okay, so you also heard about COVID vaccine. They'll tell you that with the gap of 10 days, with the gap of 14 days, okay, you need to get a booster to increase or boost your immune response. This is why I put the slide here for you to understand, because some individuals might thought that, okay, I get vaccinated, so I'm protected already. So the booster, whether I get it or not, is not important. Because most of the time when we get immunized, the first time, primary immune response, your immune response is actually that it is there, but it's actually very weak. You need a second exposure to actually enhance the level of immunity, okay, to boost it to secondary immune response so that the antibody level is sufficient, is high enough to protect you from getting infection. Okay, so I think a lot of you might ask why some of the vaccine need 2 booster, some of the vaccine just need 1 booster. Okay, one very good example is hepatitis B vaccine. Okay, hepatitis B vaccine, I think a lot of people, you get the first vaccination, then you get a booster. Okay, so most of the time you do not need a second booster, but sometimes when you go to the clinic, when you do a blood test, you found that your hepatitis B antibodies are low. Antibodies against hepatitis B are low. So at this situation, the doctors might recommend you to go and get another booster dose to enhance your immune response. Okay, so this is some of the examples that I can actually give to you all to help you understand why is it sometimes you need a extra booster. Okay, this antigenic drift and antigenic shift. So a lot of people ask me about why is it a virus can suddenly become so deadly, suddenly infect human. So actually all of this can be explained through antigenic drift and antigenic shift. Okay, so we look at antigenic drift first. So antigenic drift is basically the natural evolution process of a virus. Even for human, even for bacteria, fungus, every generation they produce, there might be some mutation that occur. So this mutation might accumulate through 1 generation, 2 generation, maybe 10, 20, 100 generation. So it is more prominent to be seen in virus and bacteria because their life cycle is actually very short. For bacteria, every so-called every 20 minutes. For virus, every 20 minutes they can produce like 10,000, 20,000 of the copies. So mutation can actually happen much faster in those pathogens. So in antigenic drift, it will slowly accumulate, and to a certain extent it become very different from the original parent virus or parent bacteria. Okay, so this is antigenic drift. However, in antigenic shift, this is normally what we see in outbreak of disease. Okay, this is normally what we see in outbreak of disease. Sudden outbreak of a disease. For example, influenza. Why is it there will be sudden outbreak of like H1N1 of influenza occur and causes a lot of death? Okay, it is actually caused by antigenic shift. Okay, antigenic shift, it can happen in 2 ways. Okay, one is with the merging of this so-called different virus strain, or different pathogen bacterial strain, okay, or it can actually due to acquiring of a genetic component from outside of the organisms, okay, and they acquire new trait. For example, they can acquire new toxins, they can acquire new receptor binding sites, okay, that helps them to, originally maybe they couldn't infect human, but because they acquire these binding sites from other organisms, then they become infectious to human. So this is two different observation that we can see in infectious agent. Okay, so I think this figure will show you much better antigenic drift. Slowly, okay, from this H3N2 here, what you can see here, the antigen changes through gradual mutation. Okay, so normally this kind of condition, the immune response that the individual have against those pathogen, okay, are able to control the infection. Okay, the antigens that presence, okay, they can actually control. But in seasonal case where it change, then maybe the efficacy of the antibodies that are produced might not be useful. Okay, then we look into antigenic shift. Okay, so antigenic shift normally we see is the combination of maybe multiple particles of virus, multiple strain of bacteria or viruses, they combine and they create a new type of strain that can infect. Okay, so normally let's say for example the virus that infect the swine, okay, can also infect human. At the same time, the virus that infects swine only and doesn't infect human, both of it can combine, and the virus that infect the swine suddenly, because of this antigenic shift, okay, it become infectious to human. Okay, in this case, you will get a sudden outbreak, a sudden burst of outbreak of infection in human population. Okay, now I will bring you all into the type of vaccine. Okay, so type of vaccine that I will cover here is live attenuated vaccine, inactivated vaccine, subunit recombinant vaccine, toxoid DNA, and vector vaccine. So I'll briefly bring you all through on this different types of vaccine. Okay, live attenuated vaccine is basically you're having the pathogens, you're having the infectious agent, you're having the virus and bacteria, you weaken them. Okay, you weaken them. Okay, so the examples that I use, okay, like the one that I have done for my uh, research study. So I actually mutate, okay, mutate the bacteria and weaken the bacteria by mutating the toxin gene. So the bacteria do not produce the toxin. Okay, when the bacteria couldn't produce the toxin, it will not lead to a disease. At the same time, this bacteria still active. When it go into the human body will treat them as a foreign agent, and they will attack it and produce antibody against it. So this kind of vaccine, it is the best type of vaccine because it mimics, okay, it resembles the actual infection condition. Okay, so live attenuated vaccine is considered the best type of vaccine, but it also has some limitation. Okay, for example, in individuals that are with weakened immune system, okay, they are not recommended to take this kind of vaccine because this vaccine, they might be opportunistic pathogen. So in individuals with weak immune response, when they take this vaccine, this bacteria or virus, they might become deadly to those individuals. Okay, so for this vaccine also, because it is alive, the storage also would be very difficult and tedious. So it has to be stored cold chain at 4 degree or -20 degree. It has to be transported throughout the process, has to be cold whenever you expose to heat, you might lead to this vaccine to die and it become ineffective, not effective. Okay. Okay, this is the example of the vaccine, how we do it. So this is the gene. We look at the gene level, molecular level. We know that these are the toxin genes. CTXA is the toxin gene that produce the toxin. So we just remove it and the bacterial strain become non-pathogenic. It will not lead to death and we can use that as a vaccine candidate. Okay, so the next one will be inactivated vaccine. Okay, so inactivated vaccine is actually more straightforward and easy. So in COVID vaccine, actually there's one that is inactivated. Okay, in COVID vaccine, in the market, there's one which is inactivated. They produce the virus particle and they inactivate it. Okay, some of the manufacturer, they use formaldehyde to actually kill the virus particle and purify it and use that as a vaccine candidate. So this is very simple. You can actually use a heat inactivation. You can use a chemical inactivation to kill the virus or bacteria and use that, inject into your body and induce immune response. So most of the time, this kind of vaccine, it actually induce lesser immunity, it gives lesser protection, but it can be overcome by giving, a booster dosage. Okay, it can be overcome by giving booster dosage to enhance the immune response. Okay, over here I want to highlight to you is the flu vaccine. Okay, the flu vaccine actually needs to be given yearly because the strain of the flu virus, there's a lot of strain, so every year the strain will change. So most of the time, the vaccine that is against the flu virus for maybe year 2018 might not be effective for year 2019. Okay, and it will definitely not be effective for year 2020. Okay, so because of the antigenic differences, okay, and also the immune response that develop, okay, the immune response that develop is not long-term immunity. It might be faded throughout the time. So if you remember in the news before on COVID, they found that those individuals recover from COVID infection, okay, when they check their immune response, when they check the antibody level, they found that after 3 months, the antibody level drops. Okay, it shows that the immune response, protective immune response that developed through the infection by COVID will not sustain. Okay, that is why the vaccine that we have in the current market for COVID, most of it have booster dose. Okay, then we look into subunit and recombinant vaccine. Okay, subunit and recombinant vaccine is a much safer version because they only choose particular component of the pathogen. Okay, it can be a virus or bacteria, proteins, surface proteins, or polysaccharides, the sugar that coating the virus or bacteria. These are some of the vaccine that we can use to protect ourselves. Because we know that, okay, if you want to protect against bacteria A, you just take the surface antigen, okay, surface protein that coat the bacteria and inject into your body will produce antibody. So when this bacteria attack you, the antibody will go and attack the antigen, the protein that is on the surface, and kill the pathogen. Okay, this is one of it, and this is considered the safe version of vaccine because it doesn't have any virus or bacteria, live virus or live bacteria presence in the vaccine that we inject. Okay, so the example is the hepatitis B, okay, hepatitis B vaccine, Haemophilus influenza type B vaccine. So this vaccine, even though it doesn't provide a very long-term protection, but it helps to reduce, okay, the infection in the community. Okay, then we have toxoid vaccine. Okay, toxoid, basically they use toxin, okay, they inactivate it and use it as a target for the immune system to develop immunity. Okay, there over here you have diphtheria, you have tetanus, pertussis. Okay, these are toxoid vaccine that we get vaccinated. So in our national vaccination program, these vaccines are actually given to our kids, our younger generation, to protect them from getting infection. Okay, so now we come into DNA vaccine and vector vaccine. Okay, DNA vaccine, basically they use the DNA, the genetic materials, inject into your body and use your body to produce the proteins, the antigens that are needed for immune response. So when your body produces the antigen will be recognized as foreign antigen, foreign protein, and your immune system will produce antibody against it. Okay, this actually same goes with the vector vaccine and also mRNA vaccine. So they use your own body cells to produce the antigens. Okay, the bacteria, the virus component, so that your body can recognize that as foreign and produce antibody, protective immune response against it. Okay, so list of vaccine that are available, you can see that we have cervical cancer. Don't just restrict the vaccine scope to infectious disease. Actually, cervical cancer actually is caused by HPV virus, and this vaccine can prevent the occurrence or incidence of cervical cancer. Okay, we have cholera vaccine, we have COVID vaccine, we have diphtheria, hepatitis B, influenza. Okay, Japanese encephalitis, it is in our national vaccination program, but it is for the Sabah Sarawakian only. Okay, that regions, because it is more prominent over there. In our peninsular Malaysia, we don't have that vaccine in our national vaccination program. Okay, for the young one. Okay, we have this typhoid vaccine for all the food handler. Okay, food handler, they are required to get this typhoid vaccine. Okay, because if they don't get it, they might be a carrier for typhoid. Okay, and they will leads to an outbreak of typhoid. Okay, varicella is the chickenpox vaccine. Okay, then you have yellow fever, rotavirus. Rotavirus is not in our national vaccination program, but you can actually get this vaccine outside. Okay, outside at the private practitioner. Okay, rabies vaccine also, normally rabies vaccine will be given, okay, when you are actually exposed to rabies. Okay, these are some of the vaccines that we can see in the market. Okay, so in this schedule, what you can see is that this is a national vaccination program. Okay, so we have all list of vaccine. So from this program, you can see that the government is spending a huge sum of money to vaccinate individuals, okay, to ensure that protection is given to the young one, okay, especially babies. So it is, you can actually roughly calculate the dosage and the dose given and also the amount that the government spent. One dose of vaccine can easily cost 60 ringgit. Some of the vaccine given here, the recently one, the recent vaccine that just approved for the young one actually costs roughly around 200 ringgit per dose. Okay, so the cheapest can go up, can go as low as 60, 50, 60 ringgit. Okay, the more expensive one goes up to 200 ringgit. So you can actually estimate the amount that the government spent to ensure that all of us are protected. Okay, so optional vaccination, you can actually go for rotavirus for 6 weeks and above. It is very important because rotavirus actually, it doesn't cause death in adult, but it might lead to death in baby infants. Okay, so I will advise all of you, if you have babies, you have relative with babies, advise them to go for rotavirus vaccination. Okay, this rotavirus vaccination is very simple. There's no injection, you just drop in your nose or mouth. Okay, so pneumococcal, this is the recent one that the government added. Okay, it can be vaccinated 2 times or 3 times, depending on the brands and also the dosage. Okay, so this vaccine, one dose costs roughly around 180 to 250. Okay, the government is actually giving you for free. So for influenza virus vaccine against influenza also is important, because we also have outbreak of influenza. If you remember last year, before the COVID, we are actually attacked by influenza in the northern region of Malaysia and the central region of Malaysia. A lot of schools, kids are actually infected by influenza. Then hepatitis A and chickenpox vaccine, you can actually choose to give to your babies and to protect them from getting those infection. Okay, so these are the schedule. And okay, here we are actually looking at COVID-19 vaccines. We have Pfizer, AstraZeneca. I think a lot of individuals have been seeing the news in the news. AstraZeneca is actually having big issues because a lot of country, they are actually banning it because of the side effect that develop. Okay, so I'm not here to comment on that, but these are some of the things that we have to consider. So if really it is causing any adverse reaction, then we stop using that. Okay, so for Pfizer's, for Sinovac, Pfizer's is mRNA. It is, the principle is almost the same, just that it skipped the step of DNA. Okay, you directly give the individuals with mRNA and they start to produce the proteins of the virus and induce immune response, protective immune response against the infection. Okay, for Sinovac is a kill virus particle. So the company produced a lot of antigens, okay, virus particle, the virus, and they kill it, and they use the whole virus as the vaccine. Okay, so for Sputnik and also CanSino Bio, they actually use a vector adenovirus. Okay, they use the adenovirus, they clone, they transfer the genes that codes for the COVID-19 virus proteins to the vector and use that vector to produce those antigens against the COVID. Okay, so this is how the process work. You have protein base, okay, that just take the protein. You have this viral vector. They use a virus. Okay, you clone the gene into the virus vector and use the viral vector to produce more of the proteins. So everything will still go to the protein, the spike protein here. Okay, whether it is a protein base, whether it's viral vector, or whether it is mRNA. And this protein will make our immune system to produce antibody and protect us from infection. Okay, that's all from me.
Thank you, Dr. Tan, for sharing. We are now come to Q&A section. If you have any questions, please click the chat box button and paste your questions. Participants, kindly fill out the evaluation form which I already pasted at the chat box. Dr. Tan, I have some questions for you.
Yes.
Actually, how long can a person be protected from infections after vaccinations?
Okay, about this question, it depends on which type of vaccine that you are taking. Okay, so maybe I use an example of different component of vaccine. Okay, we have vaccine that is made of proteins. We have vaccine that made of lipopolysaccharide. Okay, so most of the time, for the immune response that develop against lipopolysaccharide, they have a lower protective duration as compared to a protein-based kind of vaccine. Okay, so this is a general kind of conclusion that we can come across for the vaccine type. That's why it is very important that for any vaccination, they need to any studies, any researcher that develop vaccine, they need to study the duration of protective period. Okay, then they will determine whether the vaccination needs to be given a booster dose or not. So you can see for COVID vaccine, okay, COVID vaccine, through study by scientists, they found that for individuals that they get infected even by this COVID virus, okay, their immune system will get boosted, their immune response is there. But very quickly, after 3 months to 6 months, those immune response drop. Okay, the immune response drop. When the immune response drop means that there are chances of those individuals get infected by the virus again. So that's why you see after the scientist studies, a lot of vaccine they are actually given with booster. So most of the booster is given after 2 weeks of vaccination because our immune system need 2 weeks to produce the primary immune response. If we go to what I've presented earlier, you get the graph. The primary immune response is actually very low. So after 2 weeks, when we give a booster, it can actually boost up the immune response to a very high level. So after the booster, normally the immune response or the protectiveness can last for 6 months and above. Okay, can last at least 6 months and above. Okay, that's why it depends on which type of vaccines that you're taking, and from the study by researchers, they can actually tell you how long your immune response can last. Okay, some individuals, let's say for example, just now I gave you the example of hepatitis B. Okay, some individual after 5 years, the baseline antibody against hepatitis B virus actually still very high. So those individuals do not need a third booster. But for individuals, when they test the antibody is low, those individuals need to get another booster to boost up the immune response, boost up the antibody titer, and this will actually ensure them to get protected from hepatitis B virus infection. Okay, so I cannot give you a certain answers on how long you get protected. Even for COVID, no one can tell you. Okay, because COVID, they start vaccination, I think, in early of this year. Okay, until now it's just 3 months. So the maximum they can tell you is that 3 to 4 months is still protective. After that, you have to go and test it out. Okay.
Okay. Dr. Tat, what is the difference between vaccine and immunization?
Okay, vaccine and immunization. Okay, a lot of people get very confused. It's basically a just English term. Vaccine is a so-called the things the component that we use, okay, to boost the immune response. Okay, immunization is actually a process that need to be that need to go through to uh, induce your immune response. So in other words, that we can say that we use a vaccine, okay, to inject into individuals, okay, to induce immune response, and this process is called immunization, or you can call it vaccination. Okay. Is there any more question?
Whether got question from participants or not? Okay, I think participants will not have questions. So, okay. Last but not least, today's webinar will be recorded at our official Facebook page, Utah for You. You may also view our past webinar at Utah Facebook page. For further inquiry, you may contact us through WeChat, Facebook, hotline, or email. Thank you for joining us today and have a nice day.